Good afternoon, good evening, everyone. I am Ruba Najjar from GenScript. It's my privilege to introduce Dr. Sergej Kudruk. He is our distinguished speaker for today's presentation, titled Advancing Gene Editing Technologies with Spherical Nucleic Acids. Dr. Kudruk is a postdoctoral fellow at Northwestern University, affiliated with the Department of Chemistry and the International Institute for Nanotechnology. He holds a Master's of Science and a PhD in Chemistry from the University of Manchester. His extensive research interests encompass chemistry, biology, and biomaterials, with an emphasis on developing therapeutic solutions for cancer and neurodegenerative diseases. Throughout his doctoral studies, Dr. Kudruk pioneered methods for precision delivery of various molecules into mammalian cells and innovative technologies focusing on nanotherapeutics interacting within the physiological microenvironment. For today's presentation, Dr. Kudruk will be discussing the exceptional properties of spherical nucleic acids, or SNAs, and their application in nanomedicine, particularly how they enhance the delivery and efficacy of gene editing technologies, such as siRNAs and CRISPR-Cas9 platforms. If you have any questions during this presentation, please type them into the Q&A box, and we will address these questions during the Q&A session at the end of the presentation. Please join me in welcoming Dr. Sergej Kudruk as he shares his insights into this promising realm of gene editing advancements. Dr. Kudruk, the floor is yours.
Good afternoon, everyone. Thank you for this great introduction, and I am extremely thankful for this invite from GenScript organizers to speak at this conference. And it's a great honor to speak alongside these tremendous leaders in gene and cell engineering. And today I would like to talk about how we can advance gene editing with technologies like spherical nucleic acids, which is a modular platform which we can apply to many diseases. And in general, the promise of gene regulation lies in its potential to precisely control the expression of genes, enabling targeted therapies for a wide range of diseases. By modulating the gene activity, we can correct genetic disorders, enhance the body's immune responses, and also suppress harmful genes that contribute to conditions, for example, like cancer. And there are a lot of advances right now in gene regulation technologies, such as CRISPR or RNA interference, and they offer the ability to fine-tune genetic expression with high specificity and minimal off-target effects. For example, antisense DNA, ASOs, or siRNA, with a small interfering RNA, are two powerful approaches in gene silencing and regulation. For example, ASOs are short, single-stranded DNA or RNA molecules that bind complementary to mRNA sequences, leading to the degradation of a target mRNA or inhibition of its translation. This method is highly specific and can be designed to target virtually any gene. siRNA, on the other hand side, involves double-stranded RNA molecules that are processed by the cellular machinery to produce short fragments that guide the degradation of complementary mRNA. Both approaches are used to reduce the expression of specific genes and offer great potential for treatment of genetic disorders, cancers, or other diseases, which by effectively silencing harmful genes and modulating the gene expression pathways. But there are also some problems with these approaches. When we just look at the bare antisense DNA or small interfering RNA, we have a problem with pharmacokinetics. They degrade or are cleared by the body very fast. And we have also a lack of cytosolic exits, since it's a very highly negatively charged components. On the other side, we have also, as a gene regulation tool, the CRISPR-Cas9 or CRISPR-Cas13, and we're very revolutionary editing platforms with distinct mechanisms. For example, Cas9 is an endonuclease that creates double-stranded breaks in DNA at specific locations, guided by a complementary RNA sequence, allowing for precise genome editing by adding, removing, or altering the genetic material. Cas13, in contrast, targets RNA instead of DNA. It uses guided RNA to bind the complementary RNA sequences, enabling the specific cleavage and degradation of targeted RNA molecules. This RNA targeting capability makes Cas13 also a powerful tool for studying RNA function, regulating the gene expression, and developing antiviral therapies by degrading viral RNA, for example. They offer these great advantages, but again, they have multiple problems. For example, the size barrier of this large macromolecule. Also, the other problem is how we can maintain activity. And also, we have a high degradation rates of a specific enzyme inside of a body. And we as a chemist, we have the tools to target these drawbacks, utilizing the assembly and nanoarchitectures of the biomolecules of our desire. And one of promising nanoarchitectures which utilize individual biocompartments in a 3D placement to achieve biological advantages are spherical nucleic acids, or in short term, SNAs. And these structures have a generalizable platform. We have inside a generalizable core, which can be PLGA, polymer particles, for example, or liposomal core particle, or protein core particle. And when we utilize this core, we can decorate the exterior of our core with nucleic acid shell. And by doing that, we have a few advantages for our biological systems. For example, we have increased stability in serum or also in the bloodstream. We have a more controllable immune response of our nanoarchitectures. We have an elevated cellular uptake by maintaining the core functionality. And this is pretty exciting. When we look at more biological cellular interactions, as I said before, our particles can be, the SNAs can be biocompatible, very resistant to degradation, and they can efficiently getting uptaken by scavenger receptor-mediated endocytosis. And here it's the modular platform is getting leveraged because excitingly we can fine-tune where these particles can go inside of the cellular organelles. For example, for using our SNAs as immunotherapeutic, we would like to activate them in endosomes to stimulate the toll-like receptor. But by modulating the nanoarchitecture in our direction, we can also achieve endosomal escape and also access, for example, to the nucleus for efficient gene editing. And as described before, the SNAs are very modular, but the old generation of SNAs showed already a lot of advantages, although we didn't introduce the fine-tuning of the nanoarchitecture. And it showed a significant potential in wound healing by enhancing, for example, for delivery of nucleic acids to target cells. It was accelerated wound closure, what you see on the upper left side, and also improved tissue regeneration in treated wounds by also modulating the reduced inflammation. On the other side, we have also some successful results where to treat glioblastoma cancers inside of a mice, for example, which is a highly aggressive brain cancer. And we saw a significant tumor reduction in animal models when we treat the animal models with spherical nucleic acids, which carry a microRNA sequence to modulate the environment. And we also we have extended survival rates in those particles. On the other side, we have also retinal diseases, which we can target. And with SNAs, we can preserve retinal structure and function in animal models and reduce the retinal degradation by also modulating the gene expression associated with retinal degenerative diseases. And last but not least, we have been successful to treat psoriasis, and we had a significant reduction in psoriasis symptoms, including decreased inflammation, and we had also reduced abnormal proliferation of these specific cells. And as I described it before, the platform is very exciting, but the old generation, as I described before, have still some limitation in escaping the endosomes. And right now, people are already utilizing different approaches to push the endosomal escape. For example, use protein sponge effect, where we use highly charged polymers of particles that absorb cytosolic proteins and facilitate the release of therapeutic, for example, nucleic acids into the cytosol. The disadvantage of these particles is the high charge density will also induce higher toxicity. On the other side, we have also membrane destabilizing peptides, and they're designed to interact and disrupt the cellular membrane, aiding to escape the nucleic acids from endosomes into the cytosols. But again, this destabilizing peptides can also disrupt the plasma membrane in other cellular membranes. And also, people are using lipid-based formulations, for example, positively charged liposomes or lipid nanoparticles, to force the escape of nucleic acids, for example, into the cytosols. But again, there's some disadvantages. For example, lipid nanoparticles have limited tissue specificity, for example, beyond the liver. Yeah, and each of these strategies has shown promise in improving cytosolic access for SNA structures and other nucleic acid-based therapies, but they also come with challenges. And these challenges we would like to address. And this reason, like in this talk, I would like to try to answer two different questions. And the first question is, can spherical nucleic acids architecture improve small strand regulators by tuning the SNA structure, and also by maintaining one defined architecture? And also, can we apply this knowledge to different cores of the spherical nucleic acids? And one of the very accessible, stable polymer PLGA nanoparticle core, which we can use for rapid DNA conjugation, is, in this case, the PLGA SNA. And we fabricate with PLGA SNA particles using the process involved in dissolving PLGA in an organic solvent, emulsifying it with a nucleic acid solution in an aqueous phase, and then evaporating the solvent to form solid particles. And after the surface modification with nucleic acids, we can observe the increased biological advantages of the spherical nucleic acids. As one of quality control of these particles, we're using, for example, AFM or TM. In this middle section, you see the AFM pictures, and that we have increased size after the functionalization of the bare PLGA particles with the DNA sequence. And also excitingly is we hit another aspect of this rational nanoarchitecture, is that by varying the length of a PLGA polymer chains, we could control the encapsulation efficiency. And also the rotation of, for example, of hydrophobic cargo, in this case, coumarin. And we see that longer PLGA chains increase hydrophobic interactions within the core, leading to the better encapsulation and also reduce premature leakage. And this tuning is also allowing for a controlled release of a coumarin, providing a sustainable therapeutic effect. So this is a very promising particles, but it's again the question how we can fine-tune it to increase the cytosolic entry. And like since already decades, calcium is known and that it can induce endosomal escape. It can release the cargo from endosomes by acting as a trigger for for disruption of endosomal membrane. And here we demonstrate that calcium ions enhance the release of therapeutic agents from PLGA SNAs by destabilizing the endosomal membrane. And the advantage of our SNAs is that the calcium is retained within the PLGA SNAs due to their acidic environment, which allows for the sustained release of calcium ions over time, promoting endosomal escape. What you see in the lower left graph, we have a reduced dissociation of calcium ions from the PLGA particles, PLGA SNA particles compared to the free siRNA, which can also complex calcium ions. And also to understand this specific nanoarchitectures, can they really escape the endosomes? And here we used the FITC DNA flare, which was used to study assess the cytosolic accessibility. We modified the specific flares with styrosol orange, and when they interact with RNA of choice, we would have fluorescence turn on. And in this confocal images, you see that we have increased fluorescence turn on when we use a T21 sequence on our PLGA-SNA particles, and which are salted with calcium. In comparison to the control sequence, which was an A21 flare sequence, which can't bind, for example, for the specific to the polyatols of the mRNA inside of the cytosols. And further, we ran T21 and A21 as a control, where we didn't induce any calcium salting, and you observed a very significant drop of the signal. So after proving that we have enhanced cytosolic access, we also look at the cellular uptake and where are our PLGA SNA particles are located. And on the lower left confocal graph, you see after determined time, we see on the left side PLGA SNAs colocalizing these endosomes. And when we have calcium salted PLGA SNAs, we have a decrease of colocalization with endosomal compartments. And this also, like after understanding how they behave in cells, we also looked how we can decrease the luciferase expression inside of the cells. And we ran the different control groups, and the main takeaway of the study was that with loop 2 calcium chloride salted PLGA SNAs, we can achieve around 94% gene knockdown, which is comparable to the lipofectamine LUC2 siRNA, which was pretty exciting. And now we're shifting gears a little bit, and I would like to discuss the second question of this talk, and can we use the advantageous structural properties of SNAs to a more complex enzyme with a defined activity, especially focusing on Cas9 protein. And like by addressing this problem, we have a multi-component issue right now, because Cas9 is a large molecule. It presents significant challenges in gene editing applications, and one major hurdle is the efficient cellular uptake of Cas9, as its large size limits its ability to penetrate the cell membrane, because once it's inside the cell, on the other side, Cas9 must also escape from the endosomes. A process, as discussed before, is often inefficient and can result in degradation of a molecule. Moreover, successful gene editing requires also a precise import of Cas9 into the cell nucleus. A step is also complicated by its large size and the presence of nuclear pore complex barriers. And these challenges highlight the need to advance delivery systems and strategies to improve the efficiency and effectiveness of Cas9-based gene editing. And already, as described before, in recent years, we already introduced the concept of covering proteins with DNA to enhance their biological advantages. And we demonstrated that DNA-covered proteins, known as pro-SNAs, exhibit significantly longer circulation times in the bloodstream compared to the non-conjugated counterparts. And also the dense nucleic acid shell protects the protein core for enzymatic degradation and immune recognition, allowing pro-SNAs to maintain activity longer in the bloodstream. And this extended circulation increases the chances of reaching target organs and sustain the activity. What you see in the middle panel, where you see, for example, a delivery or T34 covered pro-SNA, and we have a very high activity in spleen, liver, or kidney in comparison to the blank native beta-galactosidase, which was protein of choice for this specific study. And later also, we expanded this concept to explore, for example, targeted brain delivery, where we incorporated a transferrin receptor targeting aptamer, enabling the delivery of enzymes across the blood-brain barrier. And the transferrin receptor is already highly expressed on the blood-brain barriers of endothelial cells and will facilitate the receptor-mediated endocytosis of pro-SNAs into the brain. Yeah. And it's like this approach could be also a promising approach for neurological disorders to overcome the challenge of a blood-brain barrier and underscore the advantages of protein SNAs. Yeah, and when we look now on CRISPR, what is like the main focus on our talk, the CRISPR-Cas9 system requires a multi-component design to effectively transport its components to the nucleus for gene editing. And the design includes the single guide RNA, sgRNA, which directs the Cas9 protein to the target DNA sequence. Furthermore, the GALA peptides, which are used to facilitate the delivery of these components across the cellular membranes due to the ability to form transmembrane pores. The nuclear localization signal, NLS, is crucial for guiding the Cas9 single-stranded RNA complex into the nucleus. And as I described already in previous studies, an oligonucleotide shell, which can be employed to stabilize the system and enhance the efficiency by protecting our core from degradation. Yeah, and together we hoped, and it also worked at some point, that all the compartments can synergistically work to ensure the CRISPR system that it reaches the nucleus and perform its gene editing function effectively. And by attaching peptides with specific cellular localization signals to Cas9, we can direct the protein to particular areas within the cell. For example, on the left side, the crucial part is the fusion of GALA peptides, which allow for effective endosomal escape. And the fusion of GALA peptides to Cas9 facilitates effective endosomal escape due to the glutamate-rich amphiphilic structures, which structures or these peptides undergo pH-dependent folding and unfolding, allowing them to remain stable at a neutral pH and become active in acidic environments like endosomes. And in acidic endosomes, the GALA peptides unfold into hydrophobic helices that can disrupt the endosomal membrane, promoting the release of Cas9 into the cytoplasm, which ensures the proper endosomal escape. On the other hand side, we have also a challenge of nuclear localization, and this challenge we faced by looking at the NLS, which is derived from a virus which has 40 large tumor antigens, specifically the peptide sequence, which is mentioned here, PKK, which facilitate the import of Cas9 into the nucleus. And this peptide can bind to the nuclear pore receptors, such as importin alpha, enhancing the entry of Cas9 into the nucleus where it can access genomic DNA. And the binding of this NLS to importin alpha initiates the transport of Cas9 NLS complex through the nuclear pore complex. Furthermore, let's look again on this complex structure to understand, for example, the cellular uptake of a Cas9 proteinase. We attach for cellular tracking studies an Alexa Floor 647 dye to a maleimide moieties. And what we can see here, when we look at the cellular compartments after 2, 4, and 8 hours, we have a high localization with the endosomes at the beginning of the study. And after 8 hours, we have decreased colocalization between the endosomes and our particles, which shows we have a transport to other organelles or outside of the endosomes. And last not but least, the oligonucleotide shell. We need also an approach how to conjugate to the Cas9 and how we've done it. We use the express Cas9 proteins with the attached peptides, which we described before, and the functionalized the surface lysines with a dense DNA shell. And the process involves conjugating amine-modified DNA oligonucleotides to the amine residues of the Cas9 protein surface. And this formation of a dense DNA shell around the Cas9 protein, effectively converting it into a SNA structure. And the SNA structures, we are all characterizing it using UV-vis absorbance spectroscopy to confirm the presence of DNA on the Cas9 protein, as indicated here in the upper right graph by characteristic absorbance peaks. Furthermore, we also use additionally STS-page gel electrophoresis, which we employ to verify the successful conjugation of DNA to the Cas9 protein, showing a shift in the protein bands due to the added DNA. And this transformation allowed for the effective use of Cas9 in various biomedical applications, leverage its unique properties. And we look, for example, for the simple cell uptake studies, and we can demonstrate when you compare the uptake between the just blank Cas9 protein and our pro-SNA, we have a 45-fold increase in cellular uptake efficiency at time hour around 2. And this enhancement was attributed due to the dense DNA shell functionalized on the surface of Cas9, which facilitated better interaction and internalization by cells. And this showing the improved advantage of SNA. Furthermore, and this last slide, we looked also how efficient can CRISPR-SNAs achieve gene editing in different cell lines. We focused here on HuCAT, human bone marrow mesenchymal stem cells, and raw 264. And we looked at the Western blot, and the results showed clearly that the presence of indels indicating successful gene editing in these cell lines. And the CRISPR-SNA exhibited significantly higher indel efficiencies compared to unmodified Cas9, demonstrating the enhanced functionality of SNA-modified Cas9. Notably, the CRISPR-SNA achieved a gene editing efficiency up to 47% in different cell lines and targets. And these are very promising fundamental results, but we still need to understand how these constructs behave in vivo. And maybe in a few years, we can talk about it more in detail with strong, sophisticated data to discuss. But now I would like to summarize my talk. And what I would like to convey you into this talk that Spherical Nucleic Acid is a modular platform which can dictate the cell uptake and provides privileged access to a wide variety of tissue types. We can navigate through this specific structure, endosomal compartmentalization through multiple avenues and use that to enrich cell spaces with SNA therapeutics. And this CRISPR-SNAs could have a potential impact in a broad range of diseases. And the last slide, I would acknowledge my postdoc advisor, Professor Chet Merkin, and also the whole Merkin group, which is a very vibrant group of people who are very hungry to advance the nanoscale science and make it more applicable for therapeutic diseases. And also the funding agencies, for example, NIH Cancer Institute, and the Robert Lurie Comprehensive Cancer Center. And last not but least, I would like to thank you for this, for your attention, and the GenScript organizers for this glamorous event. I'm very happy to take any questions. Thank you.
Great. Thank you very much, Dr. Korke. So now that we're going on to the Q&A session, please make sure to type your question into the Q&A box. If you encounter any technical issues, feel free to also message us through the chat box or the help desk. So the first question that we have for today is, what would be the bottlenecks for the SNA technology?
Okay, yeah, great question. So as I described all the advantages of this kind of technology, there's always like some bottlenecks and disadvantages when you go again back to the laboratory. And the main bottleneck right now is scalability, because DNA synthesis, like when you synthesize it just chemically in-house, it takes a while and it's expensive. And then the scale is between 5 to 10 micromoles. So it's like a very low amount of sequences which you can produce and also attach, for example, to a CRISPR, CRISPR enzyme. Yeah, then also another thing would be also the purification. Depends which conjugation you attach to DNA, like for example, a DBCO moiety or a peptide. This also makes the purification harder. Yeah. And it's like, I think these are the main disadvantages from the DNA side, but then all the time when you link this DNA to a specific enzyme, it will be also enzyme dependent, where you connect the enzyme and the DNA, and how dense is the coverage. So this is like the difficulties from the enzyme side, because every enzyme is very unique, and we might block, for example, an active binding pocket when we cover our particles with DNA.
Great, thank you. The next question is, how would the DNA conjugation of the Cas9 protein affect its activity? And if it would be sequence-dependent behavior?
Mm-hmm. Yeah. Oh, yeah, definitely. So we have all the time when we synthesize DNA and put them on particles, it's always a sequence dependent. So the main things where you can fine tune, for example, the cellular uptake is by varying the backbone. When you go for PO backbone to PS backbone, replace like the oxygen with the sulfur, then you can push the uptake possibilities of your particles, so general of DNA, further into the cells. You can dictate which sequence are you synthesizing, like for example, T sequence, very simple, or a more G-rich sequence, which can form very stable quadruplexes, which also can foster high uptake. Yeah, so it's like, because like we have such a programmable material, we are very flexible. Yeah, how long and how specific it can interact with cells. Yeah, that's a great question. Yeah.
Got it. There's another question also regarding the sequence dependence of the SNAs, and the question is specific to the uptake. I believe that was in relation to the gene regulation in the slide where you were talking about the therapeutic application.
Mm-hmm. Yeah, like this is what was described before. So we can fine tune the sequence and make it uptake dependent. For example, introducing such sequences which have a favorite uptake, for example, in cavity-dependent endocytosis. Or when we going into the blood-brain barrier, it's like we can prepare aptamers which will phase the transferrin receptor, and so introduce also higher transcytosis rate. So it's like, yes, it's very sequence dependent. And furthermore, like depends how long your sequencer is and how sterically hindered is for the enzyme. It will also detect its activity and function.
Very interesting. Okay. I think we have a couple more minutes, so I'll keep flying through the questions. I don't know if we'll be able to get through all of them. The next one is in regards to the nucleases that are known to target SNAs. Basically the question is asking if there's any specific ones that you know of.
Yeah, it's like there are few, and which will degrade. For example, DISA. And for our specific system, when you look deeply into the structure of a pro-SNA, we don't have any redox responsive linkages, for example, or any responsive linkages. So we rely a little bit on the endosome and lysosome to peel the shell and make the enzyme more accessible. Yeah, it's like, for example, we had some papers where we looked how DISA will split the sequence directly at the beginning between the particle and your full sequence. Yeah.
Mm-hmm. Very interesting. The next question is specific to injecting SNAs in vivo. And if you have done so, have you looked at the tissue distribution? Basically, they're asking where would it basically go in the cell?
Mm-hmm. Okay, also great question. Right now, like the whole SNA platform, the Spherical Nucleic Acids Nanoarchitecture platform, started as an immune therapeutic. So at the beginning of the slide, where I said, okay, at the beginning it was for immunotherapy, and at some point it developed also for gene editing. Mm-hmm. And for the specific right now, what we're doing as an immunotherapeutic, we do a subcutaneous injection and also IV injection. For the subcutaneous injection of these small SNAs, we have like lymph node targeting, like depends on the sequence, and liver accumulation. So this is from the subcutaneous side. For the IV side, not like we never injected CRISPR right now, state of the art as I close the talk, but we injected some other proteins covered with DNA. For example, beta-galactosidase. And we have a distribution across the organs, but also a more enhanced localization in the brain. So we can push it in direction, but we can't push it completely right now.
Mm-hmm. Wow, okay. The next question is specific to where you were presenting about the solid nanocores for the nucleic acid delivery. And this individual is trying to understand the emulsion of the solvent evaporation for the PLGA nanoparticles for the surface conjugation. So it looks like the question is, were the nucleic acid surface conjugated onto the PLGA during the emulsion evaporation method?
Mm-hmm. Yeah.
And if so, sorry. And if so, were the nucleic acids soluble in the organic solvent?
Okay. Mm-hmm. Yeah. So how we solve this issue? We incubate our PLGA polymers with PLGA azide small polymers. This reason, like we create a core where we have azide moieties around, like with a PEG unit and azide shell. This azide shell we're clicking with DBCO DNA. Yeah, and which solvent are you using? We're using acetonitrile as a precipitation method, and the DNA is soluble. Yeah. For example, besides this conjugation, when we would like to attach more hydrophobic moiety, we use like water DMF or water DMSO mixtures, and it's working, and the DNA is stable. Like at least DNA, RNA is like a completely other topic. Like it's very sensitive.
Very, very interesting, Dr. Kudruk. Unfortunately, we're out of time at this point. However, if you have any additional questions, please go ahead and still pop them into the Q&A box. We will try to address them as a follow-up. Thank you, everyone, for joining us today, and certainly Dr. Kudruk for a very insightful presentation.
Thank you very much. Pleasure to be here. enjoy the conference.
Thank you. And for everyone, please don't forget to visit our booths in the exhibit hall. And then, of course, please stick around for the next session. Thank you.